So far, we have been discussing about carbohydrates and its classification. We also understood the properties of these carbohydrates, including the monosaccharides to polysaccharides, and we also discussed about the important metabolic pathways in which this carbohydrate takes part in. So now we move on to the second group of biomolecules, that is proteins. So we all are aware of the importance of proteins in any living organism, and these proteins, they are made up of the building blocks called amino acids. So in this presentation we are going to focus on this amino acid and its classification. Okay, so before we move on with this amino acid, we have to understand about this protein. And this protein, it's present in most of the structures which is present in our body, not only in the supporting structures but also in the most of the hormones, enzymes, antibodies, transporter proteins, muscle fibers. They are also made up of this protein. And also we can see even in animals, animals like rhinoceros, its horn is made up of protein, and the spider web, it's made up of a protein. Even our hair, it's made up of a protein called keratin. Okay, so this protein is present everywhere in a living organism, and they are also going to be the final product of translation. So we are already aware of this DNA replication, transcription, and translation. So during this translation, the triplet codon is one in which the triplet codon codes for a single amino acid. So it's going to be very unique for every organism, and the final product is only going to be a sequence of amino acid, which is nothing but a protein. Okay, and here this amino acids, if you consider it, its structure is very simple. Okay, it consists of an amine group, a carboxyl group, and a side chain that is denoted as R. And the constituents of any amino acid will be carbon, hydrogen, oxygen, nitrogen, and there are two amino acids which also contain a sulfur group. And so far 20 amino acids have been categorized, because even though new amino acids, one or two new amino acids have been identified, only it has been restricted to 20 because the codon, it codes for only 20 amino acids. Okay, so it appears in different combination and sequences in protein, and the 300 proteins have been identified, and all these proteins are made up of amino acids which is going to be linked by covalent bond. Okay, and the very first amino acid to be discovered was asparagine in the year 1806, and this name asparagine, it has been derived from its source, that is asparagus, from which this amino acid was discovered. Similarly, the last amino acid to be discovered was in 309, which in the year 1938 it was discovered. Okay, similarly we can also say that few more amino acids have been named based on the source from which it has been identified. For example, tyrosine, it's identified, it was discovered from cheese, and cheese, it means, tyros, the word tyros, it means cheese. Okay, and also glutamine, this amino acid has been isolated from wheat, gluten, and hence it has been given the name gluten. Okay, so next we move on to the structural features. So here, as I told you before, it is very simple structure in which you have a carbon atom, alpha carbon in the center, and to which all these side chains are attached. Okay, so one carboxyl group is attached, and then we have a hydrogen and a side chain which is going to be alkyl or aromatic or whatever it is. Depending upon this R group, these amino acids have been classified, and amino group is attached to this alpha carbon. Okay, so the structure is a very simple structure, or the general structure. Okay, so here all the amino acids are designated as alpha amino acids, and the numbering is based on the carboxyl carbon. Okay, the carbon which is present in the carboxyl group, that has been given the first carbon position, and also there is a 3-letter abbreviation has been used for all these amino acids. For example, if it is called, if the amino acid is glycine, the 3-letter code will be GLY. If it is alanine, it will be ALA. Okay, so similarly for all the amino acids we have 3-letter abbreviation, and also 1-letter code is also there. And depending upon the side chain, the property of the amino acid differs, and this R group, it is going to determine the size, solubility, structure, and the electrical charge of these amino acids. Okay, and these amino acids, they exhibit this property of isomerism, just like what we saw in carbohydrates. So here if you can see, it has been classified as L and D amino acid. Okay, so in if you remember, in carbohydrates, this L and D, we have this position of OH, which is used for this categorization, right? So similarly here, this NH3 group position, it's used for this categorization. So if the NH3 is present on the left side, then it is called L-alanine, and if it is present on the right side, then it is called D-alanine. Okay, and in carbohydrate we have seen that most of the sugar molecules will be in the D conformation, but here only L amino acids are present predominantly. Okay, so now we move on to this classification of amino acid. They are classified based on the property of this R group. Okay, so the side chain alkyl group, it's going to determine the classification, and this R group, it determines the polarity, okay, of the amino acid at the biological pH. Okay, so the biological pH is 7. So at that pH, how this amino acids are going to interact with water, that is going to be one of the important criteria for this classification. And the number of amino and carboxyl groups is also taken into consideration. Okay, so the first group of amino acid is nonpolar aliphatic R group. Okay, so here the R group, the alkyl group which has been attached, it is going to be a nonpolar and aliphatic. So nonpolar in the sense it's going to be hydrophobic. Okay, it exhibits hydrophobic interaction with other amino acids in the protein sequence. Okay, so the amino acids in this group include alanine, valine, leucine, isoleucine, glycine, methionine, and proline. Okay, so each of these have their own unique properties. So if you see here, these 4 amino acids, they have exhibit very high hydrophobic interaction, and glycine is one of the most simplest amino acid. Okay, because it's an amino acid in which the R group is also going to be a hydrogen. Okay, so here if you see, this is an alanine, right? So here in place of this alkyl group, we have a hydrogen over here. And there is one more thing. All these, most of the all the amino acids, it exhibits, it has a chiral center. But this amino acid glycine, it doesn't has a chiral center because of the 2 hydrogen groups. Okay, here only if all the groups attached to the carbon, if they are going to be different, only then the particular compound will exhibit the property of chiral compound, right? But here you have 2 hydrogen atom which is attached to one single carbon. So glycine, it doesn't has the property, or it doesn't has a chiral carbon. And then this methionine, it's as I told you before, there are only 2 amino acids which contain sulfur group, and methionine is one of the sulfur containing amino acid. And then proline, it's one unique amino acid in this group because it exhibits a cyclic structure. Okay, so this cyclic structure is called as the imino group. Okay, so here we can see this is the structure of our glycine. So as I told you, we have 2 hydrogen groups here. So this is not a chiral carbon. Okay, and then we have the alanine amino acid. So here in place of hydrogen we have a alkyl group. And here in valine, we have 2 methyl groups attached to this alkyl, that is to this alkyl group, and that is valine. And then we have leucine. Okay, so leucine is kind of a branched, linear branched one, and then methionine. So here we have a sulfur group present, right? And then isoleucine. Okay, so these are all the first group of amino acids. So all these things, they because of this alkyl groups, it is going to be highly hydrophobic in nature. Okay, and this is proline. Here is the cyclic structure, and which is and this is called as the imino. Okay, and here this methionine, it is on the importance of methionine is it is the essential amino acid, and here this genetic code which codes for this methionine is AUG. Okay, this the triplet codon AUG codes for methionine, and it is the start codon, because we know that the start codon is the one which is going to initiate this translation process. So it is this codon which initiates the entire translation, and methionine is the first amino acid to be formed in any protein chain. So the second group of amino acid is aromatic R groups. Okay, so here we have three amino acids: phenylalanine, tyrosine, and tryptophan. And one important property of these amino acids is they have the ability to absorb UV light at a wavelength of 280 nanometer. So we have already discussed in practicals, right, the property of chromatophore, UV spectrophotometer, and how they absorb light, and scanning for wavelength. Okay, all these things we have seen. So this property is very much essential for the quantification of amino acids and protein. Okay, and again this is a hydrophobic in nature, and if you see here this tyrosine, it possesses a OH group, and this helps in the formation of hydrogen bonds with the other amino acids in any catalytic activity and all. And we have this tryptophan. Tryptophan possesses a ring-like structure which is called as the indole ring, and tyrosine and tryptophan, they are going to be more polar than phenylalanine. Okay, so phenylalanine is highly hydrophobic compared to the other two amino acids. So this is the structure of phenylalanine. So we have a aromatic ring attached to this methyl group, and then here in tyrosine we have OH attached to the ring structure, and this is the indole ring of the tryptophan. Okay. So next we move on to the third group of amino acids, that is the polar uncharged R group. Okay, so here they are going to be hydrophilic in nature. So the important amino acids in this group are serine, threonine, asparagine, glutamine, and cysteine. Okay, so the serine and threonine, they have this hydroxyl group which helps in bonding with other amino acids in the sequence, and asparagine and glutamine, it contains one more amine group. Okay, amide group is present here, and cysteine, here this is the second amino acid with the sulfur group. Okay, sulfhydryl group is present here, that is SH is present, and this cysteine, it has the ability to form dimer. Okay, so cis is the three-letter abbreviation of this amino acid cysteine, and once it forms a dimer, it is called a cysteine. Okay, so this cysteine is non-polar in nature, whereas cysteine is polar in nature. Okay, so we have to remember this as well. And it forms a disulfide bond when it forms a dimer, and this plays a very important role in the formation of covalent bonds in many proteins and polypeptides. So here you see this is the serine. Okay, and we have this CH2OH. Okay, so this helps in the formation of interaction or bonding with other amino acids, and then we have threonine. Okay, and this is cysteine. Okay, so here we have a sulfhydryl group and asparagine. Okay, so here we have one more amide group, and here again a amide group is present. Okay, so these are all the amino acids in this particular category, and this is the dimer formation of a cysteine. Okay, so these two amino acids, this cysteine, we have the sulfhydryl group over here, and it gets reduced, it gets oxidized, and when it is getting oxidized, this hydrogen bond is removed as 2H. Okay, and a bond is formed between this sulfur bonds. Okay, so that results in the formation of the dimer cysteine. Okay, so the next group of amino acid is positively charged R groups, and this positively charged of this amino acid is contributed by a second amino group. Okay, and here the amino acids like lysine, arginine, and histidine, they come under this group. And here arginine, it possesses guanidino group. Okay, and histidine possesses imidazole group. Okay, so here you see this is the lysine. So here we can find that it has one more additional amine group, and also here again arginine has two more NH2 group, and histidine also possesses additional amide groups. Okay, so this imparts a positive charge to these amino acids. Okay, so next we have the negatively charged acidic R group, and which has a negative charge at biological pH, and the amino acids in this group include aspartate and glutamate. Okay, so they possess a one more carboxyl group. Okay, so if you see here, this is aspartate and this is glutamate. Okay, they have the second carboxyl group attached to the alkyl group. Okay, so apart from this, there are also some more uncommon amino acids, and amino acids they are kind of modified. Okay, so here we can see hydroxyproline, hydroxylysine, and methyl lysine. All these things are few amino acids which are present in the plant cell wall and which imparts mechanical structure to the plant cell. And hydroxylysine is present in the cell, which is going to form a, is one of the fibrous protein. And ornithine and citrulline, they are the intermediates which is present in urea cycle. And these amino acids, they are not coded by the triplet codon, so they don't have any structure and all. And then the newly discovered amino acid is the selenocysteine and pyrrolysine. Okay, that is the 21st and 22nd amino acid. Apart from this type of classification, there is one another classification where these amino acids are classified based on the nutritional availability. So the first one is the essential amino acid. So these essential amino acids, they cannot be synthesized by the human body, hence it has to be supplemented through the diet. Okay, because it is very vital for the functioning of the they're involved in few metabolic activities, which is very much essential. So it has to be supplemented through diet. So these are the list of amino acids which come under this category, and the amino acids arginine and histidine, they are called as semi-essential amino acids. And the next one is non-essential amino acid. So our body can actually synthesize these amino acids, hence it is not essential for these amino acids to be consumed through our diet. Okay, so amino acids like glycine, alanine, serine, okay, all these things comes under the non-essential amino acid. And there is one more classification which is based on the metabolic fate, and it is classified as glucogenic, ketogenic, and both glucogenic and ketogenic. Okay, so here glucogenic amino acid, they are the ones which are going to serve as a precursor in glucose synthesis or in glycogen synthesis and all. And ketogenic, these amino acids can be used in the fat metabolism. Okay, fatty acid synthesis or cholesterol synthesis can utilize this amino acid. And similarly, glucogenic, some amino acids are involved in both the pathways. Okay, so they are like phenylalanine, isoleucine, tryptophan, tyrosine. It has been utilized in both. They are both ketogenic and glucogenic in nature. Okay, so the reference for this topic which I have taken from Leninger, and this topic is in the 3rd chapter of this particular book, Principles of Biochemistry. Okay, so with this we come to the end of this classification of amino acid. So in the next class we will move on to discuss about the properties of amino acids. Thank you.